So our next speaker is Ann Osbourn, coming from the John Innes Center in the United Kingdom. She's going to focus on plant-derived natural products and how they interact with various organisms. And there's a lot of stuff outside of the lab. There's a project that I wasn't aware of that she's a major player in called the SAW Project. It's a Science, Art, and Writing initiative, and it's there to help children learn interconnections between art and the sciences. She's also a poet, and so we thank you for coming all the way from the UK, and the title of her talk is Unlocking Plant Metabolic Diversity.
Thank you very much, and thank you for the invitation. So that was a great talk, and bacteria are great chemists. Plants are pretty good too, actually. So, in different ways, mostly. So I'm going to talk about the vast amount of metabolic diversity in plants in organisms with bigger genomes, and what we've stumbled across during our studies. So I don't need to tell you that there is a massive... Sorry? Okay. I don't need to tell you there's a massive amount of chemical diversity out there, and plants, as I say, are very, very good at it. And this slide just shows you a few examples of triterpenes that have various modifications stuck to them. They have interesting properties, so I've just labelled a few of them. And this is just a tiny, tiny snapshot of the massive amount of metabolic diversity that is out there. And these are quite elaborate molecules, but they all come from the same pathway. They just have different bells and whistles on them. And again, I don't need to tell you that the bacterial genomes are full of operons. Around, I understand, 50% of the genes in bacteria are organised in operons. And we've also heard about gene clusters, which are operons and other things as well. I'll come back to that. And of course, we have here the paradigm operon from E. coli, the lac operon. And I don't know that much is still understood at the moment, even about how operons form. It's a very interesting area. They didn't just all come in by horizontal gene transfer. They must have been formed somehow. So that's an interesting question. And the dogma, which again, as you know, is not true, was until recently that in eukaryotes, genes are generally dispersed around genomes, with the notable exceptions of tandem arrays of gene duplicates, for example, disease resistance genes in plants. But we know that's not true, and I've worked on microbes, I've worked on bacteria, I've worked on fungi. I remember in the 80s going to the Asilomar fungal genetics meeting and listening to people talking about their fungal toxin-producing clusters and wondering where they came from. And they didn't have genomes then. It was all lateral gene transfer. They could do southern blots with tox plus, tox minus strains, using the clone genes as probes. And the tox plus strains lit up and the tox minus strains didn't, so it was obviously horizontal gene transfer. And some of it is, and some of it isn't. So I ended up in this area via a rather strange indirect route, which was that I really like this fluorescent blue molecule that's produced by oat. It's produced in the roots, it's a triterpene glycoside, it's antimicrobial, it fluoresces bright blue. And I became interested in it because in the 1940s, a lady in Oxford called Elizabeth Turner had suggested that this molecule might protect oats against attack by soil pathogens. And so we set about simply to test this, because at the time, people were very focused on induced defense responses in plants, on phytoalexins, on mechanisms that were triggered in response to challenge. And these molecules just sit there, they're produced in the root as part of normal growth and development. They're antimicrobial, and I just wanted to know if they were doing anything important. So we could set up a screen. We used a chemical mutagen. We screened diploid oat, because I can't do hexaploid genetics and struggle with diploid. And we, because it was such a sensitive screen, we got nearly 100 mutants in the pathway for the synthesis of this hideously complicated molecule. And what we showed, and we published a paper in 1999 in PNAS, is that mutants that do not make the fluorescent blue stuff have enhanced susceptibility to disease, which you can see down here. This is a fungus that's a major problem on wheat. It doesn't infect oats, but if you take away this fluorescent stuff, then it does. So we proved the point that Elizabeth Turner had proposed, which was that this compound was providing protection. And we would have stopped there. Any sensible person would have stopped there because it was diploid oat. It was a hideously complicated triterpene. At the time, very little was known about triterpene biosynthesis in any plant species. I should say this molecule is particular to oat. It's a specialized metabolite that's only produced by the genus Avena. There are many other triterpenes out there, mainly in the dicots. Huge amount of diversity. But when we started this, nobody had even cloned a gene encoding an enzyme to make the simple triterpene scaffold, which is beta-amarin. But we realized that the genetics was telling us that something really strange was going on. And it seemed to be suggesting that the genes for this multi-step pathway were linked, which of course doesn't go with the dogma. So quite a few years later, having made a back library and done an awful lot of biochemistry, genetics, gene cloning, looking at enzymes, we found that the genes for this compound, which is called Avenacin, are indeed clustered. So the microbial people in the audience might be laughing when I say this is a cluster, because we have around 60 kb between some of these genes. But this is actually a gene-rich region in a plant genome. And the important thing is that the genes encoding these various enzymes shown here all encode completely different classes of enzyme. They're not tandem duplicates, they're encoding triterpene synthases, P450s, sugar transferases, acyl transferases, methyl transferases, but they're all part of a pathway. A pathway that, I can go into this afterwards if anyone really wants to know more, a pathway that has not been introduced by horizontal gene transfer from bacteria. It has evolved relatively recently in evolutionary time, and it's specific to oat. The blue fluorescent stuff is actually in the epidermal cell layer, which makes sense if it's providing protection. The genes are expressed very specifically in the roots, with little or no expression elsewhere in other tissues. And within the roots, you can see from the messenger RNA in situs here that in fact the genes are expressed in the epidermal cell layer, which is where the blue fluorescent stuff accumulates. So we have what I mischievously have called in the past an operon-like gene cluster. And when I say operon-like, it's not a bacterial operon. It's not a polycistronic transcript. These genes, as far as we know, are all transcribed independently, but they are physically linked and they're co-expressed and they contribute to a common pathway, which is required for plant defense. And there's interest in taking this pathway, working out what's missing in other cereals such as wheat, and engineering other cereals for production of protective molecules. And this is something that we're doing. It's ridiculously ambitious, but we now have a whole toolkit of genes and enzymes for triterpene synthesis from oat and from other plants. We can make triterpene scaffolds. We can modify them very selectively using P450s in ways which the chemists can't do. And then we can stick other things on there. So I now have a 5-year project to engineer wheat as one of the things that we're doing, to see if we can make it resistant to this root-infecting disease called take-all. Why should genes be organized in clusters in eukaryotes? Well, it depends what your background is, what your first assumption might be. The molecular biologists would say, well, physical clustering will allow for higher level regulation at the level of chromatin. And we certainly have good evidence that that's the case. This is some fairly early DNA fish that we did, looking at 2 genes in the oat cluster, in green and red, where Ava Vagel, a very talented postdoc who did this work, could look at she could detect these genes in the epidermal cell layer where the cluster is active, and in the cortical cells where it is not. And she could show that these genes are more strung out in the cells in which the pathway is active. And she could actually quantify this. Now, this is just descriptive. It doesn't prove anything. But it's consistent with the notion that chromatin level regulation may be important. And we've certainly gone on with other plant metabolic clusters, which I'll come to in a minute, using Arabidopsis. And we now have very good data about the ways in which these clusters are regulated. Interestingly, though, we can take the promoters out of the oat cluster and hook them up to reporters and put them into other species such as Arabidopsis and rice. And here you can see the fluorescence in oat is in the root tip and also in the lateral root initials. And when we look at promoter-reporter constructs, we can see GUS in the root tips and in the lateral root initials, both in Arabidopsis, in rice, in medicago, and we now also know in a number of other major cereal crops. So whatever's happening, these promoters have sufficient information to behave when put into other species. And interestingly, this oat pathway, which has evolved relatively recently in evolutionary time, appears to have appropriated some sort of ancient developmental regulatory mechanism that is intimately associated with root development and that's conserved in monocots and dicots. And for a developmental biologist, there's actually not a lot known about the very, very early stages of development in terms of processes that are common to both monocots and dicots. And we're now going to liverwort and more primitive plants and seeing what happens there. So we've done a lot of stuff with triterpene synthesis, and I'm not going to tell you about it, but this is just a summary of an awful lot of years' work. I think something a little bit funny's happened. I thought I saw resveratrol written on that slide then. But we know a lot about not only the enzymes, but where the enzymes are in the cell. We know that the finished product goes into the vacuole, and we know a little bit about what happens in the vacuole. One of the things that's really helped us in terms of understanding triterpene synthesis is a very neat system that was developed by George Lomonosov at John Innes. And he accidentally discovered this based on his work with Calif mosaic virus. And what he found was that if you took a little bit of 5' and 3' sequence from the RNA2 gene of Calif mosaic virus, this gives elevated protein production. If those sequences are engineered either side of a gene of interest, a coding sequence of interest, put into an agro expression vector and infiltrated into Nicotiana benthamiana leaves, you get a huge amount of protein. And he accidentally discovered this with GFP. I should say this doesn't involve viral replication, it's just a standard construct using little bits of viral sequence. And he then went on to show that this worked with antibodies, he could make empty viral particles, and we've been using it to express single and multiple enzymes in triterpene biosynthesis. It's great, you can put it all into Golden Gate assembly or whatever assembly you want to use. You put your constructs in, squirt them into the leaf, and you get the results in 6 days. It's really, really quick. And Nicotiana benthamiana is very obliging in terms of what it can provide. You don't have to worry about reductases or precursor supplies and all kinds of other things, transporters. It has an awful lot already there. So here, just to show you an experiment, this is a GC trace of an empty vector-infiltrated leaf. Here we have a trace from a leaf infiltrated with an enzyme that makes a scaffold for a triterpene, and there is a peak of triterpene here. We had a problem P450 that we knew was in this pathway. We thought that the P450 was likely to modify the triterpene scaffold. We co-expressed the P450 with the triterpene synthase down here. This peak goes down, we get a new peak. We can easily purify tens of milligrams of molecule and determine the structure by NMR, and in fact show that this is a really interesting P450 that does 2 modifications to the triterpene scaffold. So we're using this in various ways to test individual components, to build up pathways, and to do combinatorial biosynthesis, which incidentally was a term, I understand, invented by David Hopwood, he told me. I quite like that when I asked him. Coming back to the dogma, it's certainly well known that genes for some plant secondary metabolic pathways or specialized metabolic pathways are dispersed in the genome, and this is a very good example. These are the genes. This is anthocyanin biosynthesis in maize, and anthocyanin biosynthesis genes are used as markers when looking at segregation in maize genetics. And there are other examples as well, the most notable one being glucosinolate biosynthesis. But we had a question. We found this cluster in oat. There was one paper that had been published previously, it's now about 15 years ago, by Alphonse Giel and co-workers in Germany, where they'd shown that there was a gene cluster for the synthesis of a different class of defense compound in maize. And this is a cluster for the synthesis of hydroxamic acids, known as DIM-BOA and DI-BOA. They published a paper in Science. They had 5 genes in their original paper. The first gene encoded a tryptophan synthase alpha homologue that catalyzed the first step in this pathway. The other 4 genes were all very closely related P450s, but as the gentleman over there said, in this paper they showed very nicely that each of these P450s catalyzed a single specific step in this pathway. It was a beautiful paper, but I think a lot of people thought tandem arrays, P450s, and didn't really think about it that much. Beautiful paper. Then several more papers from that group and from other groups demonstrated that this cluster in maize was in fact bigger, and other genes were found that encoded different classes of enzyme. So we found the oat cluster. We then thought, well, we'll look in Arabidopsis and see what's in there, because the genome was available. It was quite a few years ago when we started doing this. So we looked for candidate gene clusters, initially based on regions of the genome encompassing triterpene synthases or predicted triterpene synthases. And we found that of the 12 triterpene synthase genes in the Arabidopsis genome, around, I think 4 of them are in regions that look like candidate metabolic gene clusters, because they have neighbouring genes that are co-expressed and that have predicted functions in secondary metabolism. So we predicted several, we validated one, this is the first one, and we showed that the genes, the enzymes in this, or these adjacent genes, 4 genes in a row, encode the components of a pathway for the synthesis and modification of this triterpene. So this then was the first example of an operon-like gene cluster in Arabidopsis. So people started to be a little bit more interested in it then. We then predicted another cluster in Arabidopsis, also for triterpenes, but it had evolved largely independently of the cluster I've just mentioned. And we have a conservative model here of how these clusters may have arisen. So a generous interpretation could be that there was a founding ancestral gene pair, but we're not confident of that. Nevertheless, since then, each of these gene clusters has acquired different genes for different enzymes, and they make different types of triterpene. So we can start to look at more than one gene cluster. So I mentioned that these genes are co-expressed, which of course is what you would expect of an operon. So in oat, we know that the genes are expressed in the epidermal cell layer and they're developmentally regulated. Here is an example of one of the Arabidopsis clusters, where we have the 4 genes in the cluster and then the flanking genes either side. And these are just different expression conditions. And you can see that the 4 genes have very similar patterns, but as soon as you get to the edges, this pattern breaks down. The flanking genes do not have predicted functions in secondary metabolism. So we're starting to learn about windows that may be new gene clusters for new pathways and chemistries. Co-expression then is one potential benefit of physical clustering. Clearly, so these pathways are making compounds which we believe or have shown to be involved in protection against abiotic or biotic stresses. And so keeping beneficial combinations of alleles together when they make a useful end product, one would imagine would be a good thing from a genetics perspective. We also have some evidence if you interfere with the integrity of these clusters by mutating them or overexpressing the components of the clusters, then you can have deleterious things happening. So in oat, for example, this mutant accumulates the blue molecule minus a phenyl sugar. The roots are really sick. We see deposition of callose, which we think is an indication of toxicity. Membrane trafficking is messed up. But if we combine that mutation with a mutation in the first step in the pathway, then the intermediate doesn't accumulate and the normal root phenotype is restored, which suggests that the effects that we see are due to accumulation of a toxic intermediate. In Arabidopsis, we also see effects on growth if we interfere with the clusters that we're working on. Okay, so there are lots and lots of questions coming out of this, and one of them is how do new clustered metabolic pathways evolve? It's like saying, where do operons come from? And so if one imagines that a gene cluster has a gene encoding what I would call a signature enzyme that makes the scaffold of the molecule of interest, and then by analogy with the microbial field, we have genes encoding enzymes that will tailor that scaffold, then it's interesting to think about the sequence of events that might have occurred and what the pros, cons, or neutral outcomes would be of acquiring a new function, acquiring a new gene during the process of assembly of this pathway. One of the interesting things we found, I've given you examples of what I've referred to as toxicity. It might be safer to say bioactivity, but we've found rather more subtle effects as well. And I just want to give you an example of that. So looking at our oat mutants very carefully, most of the mutants have normal roots, but they don't make the blue fluorescent compound, so they're non-fluorescent. But when we look very carefully at these roots, by growing seedlings on large square dishes, we found that mutations affecting the second step in the pathway led to shorter roots. So these mutants are accumulating the first committed pathway intermediate, which is a simple triterpene, beta-amarin, which is in fact common in plants. It's probably ubiquitous. And surprisingly, we found that when we look very closely at these roots, here we have the wild type, here we have mutants blocked in the first step in the pathway, which have normal length roots, and here we have SAD2 mutants, and they're super hairy. And this mutant here is a partial SAD2 mutant, which is partially super hairy. And what we found after a lot of analysis was in fact that, so we can detect beta-amarin at very low levels in the wild type, it's there. It's not all being converted to the blue fluorescent molecule. But if we accumulate elevated levels of beta-amarin, then what's happening is there's a change in sulfate as the epidermal patterning is laid down. So this is taking place very early on, and more cells are being designated as cells that will become root hairs. So this is very interesting because there's quite a bit known in Arabidopsis about genes required for root hair formation, but this is way above that. This is determining which cells are going to be the shorter trichoblast cells that will give rise to root hairs, and which ones will be the longer atrichoblasts. So this is very early in cell fate, and it hints at a possible role for these small molecules in doing something in plant growth and development that is not just toxic stuff. This figure is from a review we published in 2011. So I've mentioned the oat cluster. I've mentioned a couple of Arabidopsis clusters that we've worked on. Ruben Peters' group and Professor Yamani's group in Japan have characterized a couple of clusters for diterpene synthesis in rice. I've also mentioned the maize cluster. And here we've got the coding. So these are the signature enzyme genes and the tailoring enzyme genes. So people were beginning to think, well, okay, yeah, there's 5 or 6 of them, and it's interesting, but it's not the norm. And I'm not saying I know what the norm is, but this is a figure from a paper a review we've just published. And the number of examples of characterized clusters is growing. So we've had papers coming out over the last year or so, gene clusters for the synthesis of medicinal alkaloids in poppy, steroidal glycoalkaloids in tomato, cyanogenic glucosides. And when we look at the kinds of plants and the structures of the molecules, you can see that this is happening in very different types of plant species and for very different classes of compound. Now, so I can show you this histogram and say the number of examples are increasing. I can also say that so far, in most cases, it's not that we're just finding more examples of clustered pathways because the genes are clustered. So far, most of these have been found serendipitously. I mean, we've mined for a couple of the Arabidopsis clusters. Now we're positioned to find a lot more clustered pathways by building on the fact that we know that in at least some cases they're clustered. But we know very little about most of plant metabolism. For most of the pathways or the chemicals that you can think of, just think, do you actually know where the genes are? Do we know for Taxol, for artemisinin, for all of these medicinal compounds? A lot of the sequencing effort has gone into transcriptomics. We don't know where the genes are. So it's going to be interesting to find out how many pathways are clustered, how many are dispersed, whether there is any rhyme or reason to that. Coming back to as my starting point, really, we know that clusters are not, metabolic clusters are not particular to plants. I mean, you've heard about some beautiful examples from Michael. I've worked on fungi for years. The fungal genomes, which of course are eukaryotic, are riddled with gene clusters for specialized metabolism. And in Streptomyces, those clusters are they're not single operons, they are clusters of genes and operons. So what's going on? And are these things parallel? Are there commonalities? There are strategic benefits to the clustering phenomenon, as we know. We can mine genomes for pathway discovery, but plant genomes are bigger, somewhat more complex, and we're dealing with large intergenic regions. And Michael also mentioned waking up silent clusters in microbes, and of course this has been done in the actinomycetes. Sleeping clusters have been woken up with drugs and mutations, and we're now finding that in fact we can do the same kind of thing in plants. So there are a lot of questions around these clusters. Do we need to have them as strung out as they are? Can we edit them down? Can we make minimal gene clusters? Many of the clusters that have been discovered are for pathways that determine traits, such as disease resistance or taste or production of a medicinal compound. So it would be very handy to be able to make more compact clusters so we can move them around and modify them in plants, also in yeast, perhaps other systems rather more easily. And of course, another interesting possibility is to use the parts that we're characterizing to make synthetic clusters. So we have a beautiful set of promoters from oat that drive expression in the epidermal cells of the root tip and the lateral root initials. People are now using those promoters, which share very, very little sequence similarity, to drive the expression of other genes of interest, for example, for engineering nitrogen fixation. Another really interesting thing is that we can step back and start to look across multiple genomes, and this is work done by Alexander Butanov, who I've been collaborating with, who's mined 14 sequence plant genomes for all terpene synthases, all P450s. He's shown that there's a very clear skewing. There's a tendency for terpene synthases to be with P450s. There's a skewing within the P450, so there's a tendency for certain families of P450 to be in those pairings. He's pulled out all of the clusters we know about, which is only a small number, but there's also a ton of other stuff in there. And that in itself is a massive, massive starting point for synthetic biology and other studies. So very briefly, as you heard, I do a few other things as well. And I just wanted to take the opportunity, given that Eddie said I could say whatever I wanted, just to highlight the work that we've been developing in schools, which I started a number of years ago now. And we know that science is very beautiful. We have very beautiful images from science, and I'm sure from genomics I've seen some very nice JGI ones, for example. And these images, or carefully chosen images, can be used to underpin the introduction of a theme. For example, this is a project developed by two people in my lab. We work on plant natural products. They pick these various images out as part of a plan to work with a class to do some practical science on extracting products and analysing them. And so here we've got a mint leaf. You can see the glands on the surface of a mint leaf, the surface of the tongue, because of course that connects with the mint leaf, a lavender leaf, which one girl said looked like a pumpkin in an enchanted forest of thorns, the inside of a nose, which the boys liked a lot. I shouldn't stereotype, but that obviously goes with the lavender leaf, and a lot of interesting poetry came out of that from the boys. A mosquito piercing human skin, but that's okay because artemisinin is here to protect us. And then we have pigmentation in a leaf. We have an aphid, so the chemical defences, obviously a very important point. And then these beautiful crystals are taxol crystals. So by having a discussion with the class about how and why plants make different kinds of chemicals and introducing these images during the process and getting them brainstorming and thinking, gets them really fired up, and they brought things in to analyse. They did practical science. This was a one-day project. We'd also brought a poet and an artist into the project, so the next session was poetry. And here you can see some poetry from quite young children, from Lloyd, age 8, Science Knows. As I crush the molecules with my fingers, cells explode like a bomb. Chemicals zoom upwards into my nostrils. Strange smells make my brain dance. And then from Olivia, Spreading Colours. Colourful flames, the light looks alive, spreading slowly across the paper. Multicoloured molecules, chemicals mixing together, sparkling in the light, dancing in the alcohol. And I think you can see that they did the science before they wrote the poetry. And then for art, the artist decided out of the images that we'd chosen and from the theme that he wanted to make artemisinin. So I've shown this slide to Jay and the guys at Amaris and said, it's okay, you can stop now. Because they made a whole classroom full of artemisinin. So if you want to find out more, there is a website, or just email me. And if you're interested in setting up a project, be very, very happy to help you. Thank you. And many people have been involved in this work, and I'd just like to acknowledge all of them.
Any questions? So I was curious still in the evolutionary history of these clusters, and is it species specific, or can you find orthology and synteny in other sequence genomes?
So this is a challenge for the genome people, because this is where a lot of the synteny is breaking down. So these clusters, we don't know enough yet about multiple genomes, but I think, as is the case in fungi, you have dynamic regions of genomes. In fungi, they're often subtelomeric. In plants, some of these are subtelomeric. Some of them are in regions that are highly enriched in transposable elements. So I think it's more challenging.
Okay, one final question.
In fungi and bacteria, largest families of secondary metabolism genes are polyketide synthesis and non-ribosomal protein synthesis. Are they also predominant in plants? No?
So the examples of clusters that have come out so far are for different types of terpene, and I should say terpenes are very diverse, and different classes are synthesized in different ways. Cyanogenic glycosides, alkaloids. So for the polyketide synthases, we haven't looked. I don't know. I mean, a big thing now is devising strategies for mining genomes for all genes implicated in all classes of enzyme for secondary metabolism, and then looking at the association between them.
Okay, thank you once again.